Greetings and welcome to EHA Unplugged, the official podcast channel of the European Hematology Association, EHA.
Hello, I am Isabel Olivera, a medical writer for the European Hematology Association, EHA. Welcome to this episode of EHA Unplugged. We are going to revisit today the contribution of Professor Ivo Touw to the field of hematology. Professor Ivo Touw is a full professor at the Department of Hematology at the Erasmus Medical Center in Rotterdam, in the Netherlands. Welcome, Professor Touw.
Thank you.
First of all, I want to congratulate you for receiving yesterday the EHA David Grimwade Award that honors the career of leading hematologists in active and basic translational research. We have invited you to learn more about why you received this award and to go through your career as a hematologist. My first question is then, what inspired you to pursue a career in hematology research?
Okay, thanks. Thanks for your nice introduction and words. So, first of all, I have to say that I'm not a clinical doctor, professor. I'm a biologist. And I've been interested in biology in general. So that was my education. And then I became interested in tumor and tumor immunology when I did an internship at the Dutch Cancer Institute in Amsterdam. And from then on, I got interested in trying to understand cancer, how it develops. And I was lucky enough that I could actually get a position in Rotterdam working on leukemia. And initially, I did my studies on acute lymphoblastic leukemia. And we were trying to understand what was going on in these cells, how they grow, how they respond to certain therapies. So we were trying to culture these cells, which was really very difficult at that time because there were no growth factors being cloned. It was very complicated. And then my first step was actually that we could grow those cells with certain growth factors, particularly the growth factor interleukin-7 that was just discovered at that time. And then doing those studies, I became more and more interested in how these growth factors work, how they actually interact with molecules on the cell membrane, the so-called receptors, what is happening inside the cell. And that's actually in the early 1990s when I did this. And even before that, in my PhD, which was actually finished in... PhD was a bit earlier, but on that same topic. But at that time, we were talking 1986, some time ago, none of these growth factors was actually known or cloned. So I took it from very early primitive studies into more advanced studies when the growth factors were cloned, when we could do the studies, when we could also use molecular biology technologies. And from then on, I switched at a certain point, and this is where I got the prize for yesterday, into another disease, and this is severe congenital neutropenia.
Did your research focus first on severe congenital neutropenia or on G-SCF signaling?
So that really got hand in hand, and I can tell you how that worked. So we were doing in Rotterdam studies not only on acute lymphoblastic leukemia, but also on acute myeloid leukemia, together with my colleagues Ruud Delwel and Bob Leunberg. And again, as I was saying, the growth factors were just identified, and we were intrigued by studying what is happening with the leukemia cells, how do they respond to growth factors. And we figured out that at that time there was diversity in the leukemias. Some of the leukemias responded very well to growth factors, others did not. Some of them just underwent a proliferative response, so they grew with the growth factors, and others did grow and also could differentiate somewhat. That was relevant also for therapeutic reasons, as many people are now studying differentiation in the context of therapy. So at that time we were really intrigued, and I was intrigued, how does that work? How can a growth factor in some cells only induce a proliferative response and in others proliferation and differentiation? What's going on there inside the cell? And what is the receptor doing there? Can we really discriminate different functions of the receptor depending on how the receptor signal? How does this work structurally? So that was actually how I came into the world of the G-CSF response and the G-CSF receptor. So at that time, it was really very difficult to study this in primary cells. So I was interested in developing models, and for that I needed molecular biology technology, particularly making constructs that we could use for the receptors and also for mutants of the receptors, make constructs that we could use to express these molecules into cell models. And for that, to achieve that, I actually did a fellowship at the National Institute of Health in Bethesda with Art Nienhuis. And he was really pioneering on gene therapy and also making vectors to introduce molecules like receptors into cells. So I did that, and that was in the late 1980s. And I came back to Rotterdam with the toolbox, with the knowledge of the technology.
Was that your postdoctoral research?
Yeah, that was a postdoc. Yeah, and when we came back, actually, we started working with these new technologies, and then we were getting interested in this disease, severe congenital neutropenia, because the G-CSF response in these patients is very low, although you could give them G-CSF therapy and they would respond. So we were really intrigued by that observation. So, as I was explaining yesterday, we had a Chinese PhD student, Fan Dong, who came into the lab in the early 1990s, and we started working on this idea, this concept, and he actually was the first together, so in our group, to discover that there was actually a mutation in the G-CSF receptor that led to the truncation of a region, the C-terminal region, that was very important for the function of the receptor. And it was also linked to patients who developed leukemia. So that was our first key observation in this work, published in the New England Journal in 1995. That fueled all of our and my career basically in this direction.
Is this the discovery for which you have received the Dutch Research Council NWO Pioneer Award?
So that was at that time a very prestigious grant from this Dutch organization. And there actually we wanted to embark on making the mouse model, as we did, of course, in that program, and really to understand what is the contribution of these mutant receptors in the biology of neutropenia, and also what is the role of these mutations in leukemia progression, as we had seen that these mutations were particularly there in cells from the patients when they had undergone leukemic transformation.
Just to summarize, patients with SCN receive lifelong treatment with G-CSF, which alleviates their neutropenia. At the same time, this treatment triggers mechanisms that may lead to malignant transformation. Are these mechanisms what you have been working on in the last decades?
I'm a bit gray now, not from the work.
Yes, but could you briefly summarize the main mechanisms that you have described over the years that are at the basis of this malignant transformation?
Yes. So first of all, using the models, we were interested in how does the mutant receptor signal? What is it doing compared to the wild type receptor? And it turned out, so we started with a very simple idea. I told you that the C-terminal region is gone after the mutation. And initially we thought that C-terminal domain that is lacking and that's associated with leukemia, if it's not there, that had something to do with differentiation. So we thought maybe there are specific differentiation pathways via transcription factors that are directly linked to that C-terminal region. Now that idea in the end turned to be wrong, because we looked at this and we tried to figure out, is this really the case? And we couldn't really put our fingers on this. And then actually we realized that the dynamics of how receptors behave inside the cell could actually be very important. And I've not gone into any detail yesterday in the talk, but we have done a lot of work figuring this out. And very briefly, what is key here is that the normal wild-type receptor is activated by the growth factor. It's dimerizing, and then it gives a signal first on the plasma membrane. So PR3 kinase and RAS pathways are activated very rapidly when the receptor is still on the plasma membrane. But then the receptor is being processed. It is internalized via a very specific domain that's important for that. And then it is being degraded via lysosomal degradation. And that is actually very important for a very balanced response inside the cell to the growth factor. And it is this balance that really determines how the cells respond. So you first need a trigger into proliferation, and then in this balanced situation, what will happen is that the cells undergo gradually an arrest in G1, so they will stop proliferating, and that is then a very important step in triggering the differentiation. That process is completely disturbed with the truncating receptors, because so what we have seen in cell line models and in mouse models is that this truncated receptor stays on the plasma membrane because it lacks the critical mechanisms that causes internalization and lysosomal rooting. And in the cell line model, it is actually only giving a proliferative signal. But in vivo, the situation is a little bit different, because there, of course, G-CSF is not the only critical component driving differentiation. So in mouse model, what you see there is that you get a hyperproliferation, neutrophilia. So there is still neutrophilic differentiation going on, but clearly the signaling is disturbed. And now I come to the second part. So what we have seen in the mouse models and also in patients, the truncating mutants are really acquired during the G-CSF therapy, as you mentioned. And if that is happening, these clones can be really there for a long time. So patients get the G-CSF therapy almost daily or twice or 3 times a week to have the neutrophils up. And then these clones may appear. There can be multiple clones. Often it's not just one, but there can be multiple clones that are there. And then gradually over time, these clones may outgrow and may transform. And RUNX mutations are very, very prevalent in that. The combination receptor mutation and RUNX mutations are very frequent in this situation. And then the leukemia progresses. So that's a very important combi, the mutant receptor and RUNX mutations. But we were kind of puzzled by the fact that if you look in patients, so from our mouse and cell line data, we thought, okay, once there is a clone with a receptor mutation, and if you continue to give the patient the growth factor, those clones will rapidly grow out. So it is indeed the proliferative overproliferation that is playing a role. But in patients, that is not what we saw. We could actually see that these patients could receive the therapy. We monitored the clones, and other people did as well. And these clones did not really expand. They actually were there. We could still see that they were there, but they really did not expand. So that was puzzling to us, and that put us on the track that there must be more than just this hyperproliferation that we had seen in the patient and cell line models. And by then, so we're now talking, let's say 2010 or so, 10-ish, a bit later. Yes, so then we had this fantastic new development with the iPS technology coming into the field. And that has really helped us tremendously, because now we were able to generate a model system from patients. We could take fibroblasts and reprogram them into stem cells. We did studies on primary cells as well, but that material is rare. It's often coming from young kids because this is a congenital disease, so patients are young. You cannot always take bone marrow samples. So we were really very happy that this new technology, iPS technology, came. Another revolutionary technology called CRISPR, that we could use to introduce the mutations in the receptor and also in the other gene RUNX that I was just mentioning, to very carefully pin down what is going on in the progenitor cells from the patients if they have these receptor mutations and also RUNX mutations. And then it turned out, and this I think is something that is now being accepted also in other types of myeloid pre-malignancies, that inflammation is really a very key process that is going on. So very briefly, what we could see also by making use of these iPS models is that first inflammatory cytokines like interference, or I should say the response to interferon-induced genes program is really very high up already in the stem cells from the SCN patients. And that is completely different from what we see in the iPS models from the controls.
Is this caused by the congenital mutation?
Yes, so we have clearly seen that if you have the congenital mutations they're basically the most frequent ones are in ELANE, in neutrophil elastase encoding gene, ELANE, and in HEX. These are mutations that play a role in mitochondrial integrity and also in ER stress. But there's different flavors to that, so it is a bit more complex. So we knew already that, and we could make use of that knowledge really to particularly look in those pathways. So what we could first see is that indeed there was stress. We could see that with ROS, so-called reactive oxygen species probes, that there's elevated oxidative stress. We could also link that to antioxidant pathways. And then that actually linked, let's say, the defect, not so much to a hyperproliferation by the truncated receptor, but by inducing inflammatory stress, interferon stress, and then later also when RUNX mutations are there, other inflammatory pathways, particularly TNF and IL-6 driven pathways. So the general message is that inflammation and increasing inflammatory stress is a very important driver, we think, in the malignant transformation of this condition, driven by truncation of the receptor, G-CSF treatment, and the acquisition of additional mutations in epigenetic and genetic programs.
Do these findings open new avenues for thinking about therapy?
So that's still very early days. So we know, and still today, the treatment with the growth factor, G-CSF, is still the first treatment. So most of the patients respond well, although we have these issues of clonal evolution, as we call this. But some patients actually do not respond so well to the G-CSF therapy. So roughly, depends a bit on the genetic background and the cohorts, but you could roughly say that 85% or so of the patients, or 80%, do well on G-CSF initially. And there's also subtypes that do not respond. And obviously, you need to find new ways to treat these patients. So for two reasons. One, to avoid the neutropenia, because it's fatal if you don't treat it. And of course, to avoid the malignant transformation that you also want to. So what you want to do first is, of course, look carefully in what type of genetic subtype is there, what is the biology behind that. Is there a risk for leukemic transformation? So monitor the clones with the receptor mutations. And if the response to G-CSF is not so well, you have to find another way. And nowadays, stem cell therapy, so allogeneic stem cell transplantation, is then the first choice. But obviously that comes with the risk of treatment-related mortality. So these young kids, when they're still doing quite well on G-CSF treatment, but you see that the clones with the mutations are arising, what can you do? And this is, of course, a dilemma that I'm not facing, but pediatricians who treat these patients have this very important choice to make. What can I do? So obviously that depends on all kind of things. Is there a suitable donor, yes or no? And do I really want to take this risk of, the therapy? So that's in the field still very open. But what you hope, of course, to do is to find new treatments that avoid this difficult choice. First, of course, is that now with the new CRISPR technologies that I mentioned and the derivatives of that, you can think of repairing stem cells. So do an autologous transplant with repaired stem cells. Autologous transplantation does not have the risks of, allogeneic transplants. So that's a benefit. Obviously, this is an important development, and people are working on this. There are some technical difficulties there because the mutations in ELANE, as I just mentioned, there are about 100 different mutations. So if you want to develop a therapy to all these mutations, you have to think very carefully how to set this up. It's complex, and then of course it will take time to really, to deal with all the safety issues, side effects that may arise. But I know that people are working on this, and there's other examples of diseases, particularly in the erythroid lineage, that people are now already treating with these kind of approaches. So this is definitely a very important area. I am also thinking about other things. If you see that there is inflammation, there's drugs on the market that you can use to reduce interferon induced effects and other inflammatory signaling pathways that you can target. There's many drugs that so you might consider, once that you see those clones with receptor mutations, to dampen them and to keep them sort of silent from going into further progression by treating them with anti-inflammatory reagents. Another possibility that I discussed briefly PML and nuclear bodies, which play a role in, we think, in the G-CSF non-responsiveness in patients with certain mutation that causes misfolding of the neutrophil elastase protein. So you might also think of modulating that pathway. And finally, and that is still very early days, is that there is this pathway that is actually now being driven and also in other myeloid conditions by the expression of the so-called transposable elements. And I mentioned the example of line 1 yesterday. It's very early days, but that line 1 is playing a role in inflammation by actually, so it's expressed as a small messenger RNA, but then it is copied by reverse transcriptase in the cytoplasm. Now, reverse transcriptase inhibitors are also on the market, for instance, in HIV and other type of viral diseases. So it's very, very early days still, and I'm not actually at all trying to claim that this will be the solution. But I think it will definitely give interesting insights in what you can do with already existing drugs. First, test them carefully in the IPS systems, and that's the benefit of this. We can now really take advantage of those cell lines, make them for many more mutants than we have done today. So you can make a whole panel of these IPS lines with the different relevant mutations and just start working on using those type of compounds and drugs in all kind of combinations to see whether that would actually make sense too. But what excites me really is now with all the new technologies and again, IPS and the gene editing technologies that we now have, I think there are really fantastic opportunities to do indeed basic and translational science research into this area and find indeed new mechanisms and potentially therapeutic options for these kind of patients. And again, this is a rare disease, right? But if you take this in a little bit more broad perspective, there's many of these type of conditions, so many of them that we still don't even know what the genetic background is. We are actually also now working on new families with a new gene important for the disease, and there's still a lot to learn from that. And if you take it all together, there's many of these situations that we don't know the genetics of, but are still and they're all rare in that sense but it's important to find new therapies for these patients in the end. Even if every single genetic subtype is rare, if you take them all together, we are looking at a very large group of patients and individuals having these issues. So not necessarily severe neutropenia or severe immune deficiencies, but there's many patients who suffer from sort of milder conditions, but a mild condition can still be in the end a very unwanted and harmful situation.
You mentioned briefly that you are also interested in computational biology. Could you share with us what your plans are?
Yes. Well, I must be very modest here because so I'm now currently also in the educational committee of EHA, and previously I've been involved in setting up a very nice initiative by EHA together with ASH, American Society, and that's the translational research training in hematology. And those courses are really very nice because they bring young professionals in a training setting where they can really be exposed to cutting edge science, but also meet with leaders in the field. And that has been a very successful course. And then just recently, Brian Huntley and others who were actually in the research committee of EHA started this initiative, which is called Computational Biology Training in Hematology. And that is doing similar things as TRTH, but then focusing on computational biology, meaning bioinformatics, but also advanced, now mostly also AI-driven new let's say, computational tools to understand and to integrate very complex data sets that we now have from the next generation sequencing platforms and other platforms, and integrate all this information is really key on top of the developments that I was just mentioning to really make steps forward. And that's a very intriguing field.
Could you give us some examples of using these computational approaches for advancing research?
We are making use of relatively standard bioinformatics tools to analyze our RNA-seq data, single cell analysis, interactions, so distant interactions between cells that you can quantify and qualify by using these technologies. You can integrate transcriptome data with proteome data, with patient data, with cohort data. So there's a very many different levels of information that you can actually, and big data sets that you can integrate by using computational approaches. And I think it's a very, very good step from EHA to step into this. And this computational biology training, I really stimulate researchers to have a look at this course and think about whether it would be worthwhile for them to apply for this. And this initiative is also together, not with the American Society at the moment, but with another very important organization, which is the European Bioinformatics Institute in Cambridge. And that is actually part of the European Molecular Biology, EMBL organization. And this link, so putting their expertise in computation biology and our expertise in the hematology field together, I think is a wonderful match.
So this highlights the importance of the cross-field collaborations in the era of big data, doesn't it?
And it all starts with asking the right questions. Even if you have complex data, I mean, you have to go back and go back into very sort of crucial, often quite simple questions. Why is a patient not responding to a therapy? Why is this patient sooner or later developing leukemia? And you start with the simple questions, and then you can make use of all the fantastic technologies, including all these computational tools that we now have. Because the questions are simple, but the biology is mostly very complex. And for that, I think this is really a very important step forward.
From our conversation, it is clear that your enthusiasm and curiosity still drive your work, and this has helped to advance the knowledge and improve patients' lives. I would like to thank you very much for all your amazing contributions and for sharing your thoughts with us.
It was a great pleasure doing this. Thank you.
Thank you again. It has been our pleasure. And thank you to all of you for tuning in and listening.